Welcome to BioMars. Hello everyone. Today I'm gonna talk about mitosis and meiosis. They are very important in biology, especially when the first biology course in the UC Davis is teached about meiosis and mitosis. I still remember I do not have because I do not have any biology knowledge during the high school. I haven't taken any biology course, so I'm really struggle at that course. So for this video, I will only talk about the difference between mitosis and meiosis, and also meiotic. And for the next video, I will talk about meiosis. So what's the difference between them? The mitosis is about somatic cells. Somatic cells, you can understand that basically every cell without germ-line cells. So what are germ-line cells? Germ-line cells is egg and sperm. And somatic cells is for meiosis, is for mitosis. And germ-line cell is for meiosis. So what's the difference between them? The mitosis is to identify daughter cell, and meiosis is haploid cells. So basically, you can understand 2n from 2n plus 2n in the mitosis, and in the meiosis is 2n become 4n, 4 small n. And in the mitosis, there are several phases. The first one is interphase. Basically, is the replication. We should put that here. When the interface to the prophase, because of the replication, the interface 2n, it become the prophase 4n. Here are 4 alleles. We can call it A, B, C, D, or small a, big B, big A, small b. Everything we can call it. It just 4 very easy cells. And because of the replication, it's from 2n. See, there are 2n. There is 1, and there is 1. 2n become 4n. Basically, it become doubled, and it is really important to know their name. So big A and small a, they're homologous. And for these two, all of them are sister chromatid. Homologous and difference between sister chromatids is sister chromatids basically they're totally same. Yeah, so it just replicate big A replicate a big A, small a replicate a small a. So we go to the second step is metaphase. All of these 4 cells, 4 chromosomes, they lined in the middle of the cell. See, they line up. And also, it's not the end. There will be a spindle on each 2 sides of cell, and the spindle will attach all of them. The place this spindle attached is centromere. Centromere basically is I want amplify the, for example, to small a, right? They are sister chromatid, and here, this part middle is centromere. The spindle attach centromere and separate them. So We go to the third phase, is anaphase. I will note here, this is metaphase. This is anaphase. Anaphase basically is a cell we can separate, and because it the spindle drag each a sister chromatid to left and another sister chromatid to the right, so basically it's here. Seems like I draw my circle because it is too small, but they are not attached each other. They're not attached. They are still separate. And anaphase is basically sister chromatid segregates away from each other. Sister chromatid segregate away from each other. And then we go to the last step, and also the most important step is telophase, where basically we already got what we want. Like the left part will be same as the right part, totally same. And each cell, just like what I talked before, the 2n become 2n and 2n. They are identical. So that's how the mitosis process and how mitosis make one cell become two identical cells.